So now that we know what the data looks like, the types of information that we're going to need to do our statistical processing and comparisons, I'm going to lead you through this sort of, this workflow here around the circle. So we're going to start with the data and then walk through the different processing steps, ending with our statistical comparisons. So for proteomics data, we have a little bit of a challenge. And I'll say also that most of what I'm talking about right now in this talk is also applicable for metabolomics and lipidomics data from mass spectrometry. There are some extra steps for proteomics and some steps that are specific to proteomics, so I'll call those out. But in general, it's a very similar workflow for any of those mass spectrometry data. With proteomics, we have rather noisy data, especially in comparison with metabolomics or lipidomics. In all cases, what we're getting and what we're talking about is relative quantification. And this is all for proteomics from that bottom-up approach and a global proteomics. So we're not doing targeted analyses. We're just trying to see in general what all is there. For proteomics, we have large amounts of missing values, and this will impact the types of statistical analyses and even things like PCA. Which type of PCA can we use when we have missing data? We don't want to do imputation if we don't want to remove peptides or proteins that had missing values in some of our samples. There are certain methods that we can use to still be able to look at a PCA plot, for instance. So as you've heard from Paul this morning, there's a lot of underlying biology that we need to understand in order to really utilize proteomics data. And so I'm glad that he was here to give us a foundation there. I learned a lot. I'm going to go back and listen to that, Paul. And David mentioned some tools that are available publicly that you might find helpful for analyzing proteomics or other mass spectrometry data. If you're working in R, there's the PMART-R package, and then there are two different routes basically to get to the same user interface. Here is one of the URLs for MAP, and in our closed session this afternoon, we'll walk through using MAP and PMART-R to do this type of analysis. Other options that you may have already been familiar with are MetaboAnalyst and MS Stats. All right, so David gave us a nice overview of the different data that we need in order to use this type of workflow. So I won't spend much time here other than to say, we have our peptide level information, that mapping to proteins, and then the observed relative abundances for each of our samples. We also have information for the samples about experimental groups of interest. This could also include things like run order. Sometimes we like to visualize our data in run order to see if there's any sort of effect of that order on what we're observing in our samples. So now we're going to move to this format and pre-process step. And one of the things that we always look at with proteomics data is the amount of missing data per sample. We often see up to 40% missing values in this bottom-up global proteomics scenario. And we can have very different patterns of missingness for the different samples. So each of these bars is a sample, and we're just plotting the number of missing values in that sample at the peptide level. So there's quite a range, and this is a really typical pattern that we see. This isn't anything alarming. Sometimes you might have one sample with a really high spike relative to every other sample, and that is something to look into. Maybe something happened in sample processing, for instance. Our recommendation for this type of data is that if your identification software outputs zeros when a peptide wasn't observed, that you convert those to NA or missing. And this is specifically for working with the PMART-R package or MAP for that user interface. And The reason that we keep those missing values in our data is that imputation has been shown to be very difficult for proteomics, and it's pretty easy to go wrong and introduce patterns that aren't naturally there just by the imputation method that you've selected. So if this is something that you're going to do, just please exercise caution. And there are two really great manuscripts for either label-free or that isobaric labeled proteomics data that give some guidance and things to watch out for if you're doing imputation. We also see this common pattern in label-free data where the amount of missingness is related to this average abundance that we're observing. So we tend to see more missing values, right, at these lower abundances. Another word here on imputation. So there are some common methods, but that doesn't mean that they're the best. This half the limit of detection is particularly fraught. So what I'm showing here, so these are just box plots, one for each sample. We have two different groups, which the colors show. And before imputation, we have our box plots for log to abundance. For the same data, if we do this half the limit of detection, we get a very different looking plot for those same samples. So just be aware that imputation can really change the structure of your data and influence the downstream statistical results that you obtain. Another initial processing step that we do is to transform our data to the log scale. And this is so that we can rely on that normality assumption later on when we're doing statistical comparison. This plot on the left shows box plots, believe it or not, before that log transform. We can't even see the box because the data are so skewed. And then after we've log transformed our data, we get these boxes, and we can actually better understand what samples look like. We can see that there's maybe a little bit of a visual difference between this mock group and an infection group, whereas over here on the left, we can't tell. A large part of the workflow that we go through for proteomics, as well as other omics data, are quality control filtering steps. And there are some proteomics specific filters that we run, as well as other filters that we run across the board for metabolomics and lipidomics as well. And I'll talk a little bit more about each of these on the next slides. For those proteomics specific filters, one thing that we do sometimes is remove any peptides that are degenerate or redundant. So these are peptides that map to more than one protein. And as Paul talked about, it's not always an easy task to figure out which protein to attribute a peptide to. So sometimes people prefer to simply remove those redundant peptides from their data. So that's what this figure here is depicting, right? We see this squiggly red protein or peptide, but it is part of both protein A and protein B. So we don't know where it's coming from without perhaps a lot more work. Another thing that we sometimes do is remove any one-hit wonders. So those would just be peptides that we only see once across all of our samples. There was a question earlier about, what do you do in cases where you only see something in one sample? And this is a pretty common path forward. Sometimes we receive data, so that spreadsheet that David talked about, right, where we still have reverse hit peptides in that data. And those are there because they were used for false discovery rate calculations, and we don't want to include those in our analyses going forward. We may also have contaminant proteins present, either in ways that we would expect simply from the way samples were processed, or they could be accidentally there, right? Sometimes we've seen like human contamination in plant samples, right? And that's something that wasn't intentional or purposeful, but just occurs. So you want to remove those if they shouldn't be part of your downstream statistical comparison. So if we want to go beyond those proteomics specific filters, there are a couple of other options that are used sometimes. So one of them is, instead of just removing those one-hit wonders, we might want to have a little bit more confidence in what we're seeing. So if we want to require that a peptide is seen in at least 3 samples out of all of ours, right? We could impose that sort of a filter and get more stringent with the peptides that we're retaining. Sometimes people like to look at the coefficient of variation across all of their observed molecules and impose a filter. So this gets at variability. So it might be that people are less interested in the statistical results for biomolecules that are really variable. So you could decide to remove things that are at the far right side of this distribution. Visualization-wise, it's really helpful to look at correlation heat maps of your samples. In this particular heat map here, you can see, these chunks of red and yellow. It's not surprising if you look at this tiny axis here. So these first 3 columns and bottom rows are for the control samples. So it's not surprising that they would be less correlated, right, with the infected samples than they are with each other. So this doesn't raise any alarm bells for us, and it's reassuring because it's showing us the biology that we would expect. In the plot on the right, we see our box plots, one for each sample again, colored by treatment group, and we see this green box here in the middle is lower compared to all the rest. So that's sort of a red flag that maybe we need to look at that sample more closely. There may have been something in the processing of it that if we go back and talk to the experimental scientists, they might say, yeah, this sample looked weird when we received it, or something happened to it in the processing that could explain this, and that might be a sample we want to remove from our data going forward. The PMART-R package and map both include a way to objectively identify potential outliers. And what this method does is it takes information such as the median absolute deviation, that's the MAD up there, skewness, kurtosis, the correlation, and the percent of missing values that are in each sample. And it converts that to a distance metric, which can be associated with a p-value. So you can actually say, I have a p-value cutoff. And in this plot on the left, this horizontal line corresponds to a p-value threshold of 0.001. Any samples represented by the dots here above that line are potential outliers that we should look at. It's saying that this sample looks different from all the rest based on these metrics. And then what we can do is say, okay, well, I have this one sample that I need to look more closely at. And we can look at a box plot of the metrics that were used to generate these distances. And the X's on these box plots are for this particular sample. So we can see which of those metrics it was an outlier or it looked different on. So here, the median absolute deviation, it was right there in the middle of that distribution. But for skewness and the proportion of missing values, it had very high values relative to the other samples. And the correlation of that sample with the others in its group was much lower than for other samples. So given this example, this sample here above that line is one that I would remove going forward. It doesn't behave like the others in its group, so I don't want it introducing more variability in my data. We also have a number of typical graphs that we use just to explore our data. and understand it better. So you've already seen some of those, the box plots, the correlation heat map, a principal components analysis plot is another very common one. And so this is something that we look at as well. And if we make this plot before removing any sample outliers, it can be helpful to look at where your outliers or your potential outliers fall on this plot. And that can also help inform whether to remove a sample or not. So once we've gone through and done all of the filtering that we want to do for biomolecules and samples, the next step in this workflow is data normalization. And the reason that we do normalization is to put those relative abundance values on a comparable scale from sample to sample. What normalization is doing for us is removing any of that unwanted variability from our samples so that we can really just focus on the biological variability that we're interested in. Now, that variability that's unwanted can come from lots of different places, as Paul alluded to earlier. So it could come from the sample preparation and the sample handling. Sample storage can impact variability as well. And there can also be variability introduced by the instrumentation. So there's run-to-run variability on mass spectrometers, even if you're using the same machine, right? If you run data at different times, you can have batch effects. Different machines can have different amounts of variability that they introduce. Fun part about normalization, though, is that there's not a single method that's going to be the best method for all data sets or all experiments. So this is a processing step where you kind of need to be careful. And there are some ways that we can be guided so that we know whether the normalization approach that we've picked is good or bad. Because the different normalization approaches can sometimes introduce bias into your data. And we don't want that to happen, right? We don't want to get significant results simply because we normalize one way. So these plots are showing you, so the unnormalized peptide data here on the left, we have put these little X's over those median values there in the centers of those box plots. So if we were going to use global median centering to normalize these data, we would just, so for this first sample on the left, we would take that median value and we're on the log scale. So we'd subtract it from all of the observed peptide values for that sample. If we do that for all of these samples with their own median values, we'll get this plot on the right where all of their medians are now centered at 0. So this is one common way that we normalize both metabolomics and lipidomics data. For proteomics data, this does get used quite a bit, but we have a tool available to us that we can use to help us determine the best normalization approach to use. And the reason we can use this for proteomics data and not the other two is that we have typically many more molecules in our data set with proteomics than we do for metabolomics or lipidomics. So what we're looking at here are heat maps where the x-axis provides a function that's used to get your normalization parameter. So we have like a mean value, median. So if we were going to do that global median centering, we would be in this column. Sometimes people use a z-score, median absolute deviation, or there's a regression approach as well. And the rows correspond to different subsets of your peptides that you can compute these normalization metrics on. So global median centering would mean we're using all of our peptides, all of our peptides and taking the median and using that to normalize. What we end up getting either through PMART-R or through MAP though, is a plot like this where, you know, the cell here with the dot recommends that's the top choice. That's going to introduce the least bias into your data. So once we've moved past that normalization step, we're ready to do protein quantification. And Paul mentioned a couple of different methods for doing this. And when we've got this bottom up workflow, right, we're measuring the peptides, but what we want to know about are the proteins. That's what maps to genes and pathways. So what we need to do is for each of those peptides, we need to assign the protein that it should be matched with. And there are some common methods that I've listed here that are available through the tools that I've mentioned, and they involve kind of similar to normalization. They involve some sort of a function, right? Are you adding, you're taking the sum, are you taking a median, and which of those peptides that map to a protein are you using to sum, for instance? So it could be that you're selecting a reference peptide from that set, or it could be that you're just taking all of the peptides that map to a protein. There are ways as well to account for isoforms, and two of those are listed here. There's a BP quant method as well as a PQ-PQ that you can look at the papers for. The BP quant is available through PMART-R and MAP. This graph is showing peptides that are associated with a single protein. And so you can see that we've got these solid lines here that across the three treatment groups kind of diverge from these dashed lines. So in the control group, all of those peptides abundances are very comparable. But as soon as we go to the treatment groups, you see this differentiation. So if we look across what's happening in those treatment groups, that can give us a clue to which peptide should map to which protein or which isoforms. A note about the protein quantification step in metaproteomics. Here we have the added complexity of multiple organisms in our sample. So this kind of explodes the difficulty of doing protein quantification. And this is an ongoing research area. So there are some methods out there, but it's still constantly developing. So once we have gotten our data to the protein level, we are finally ready to make our statistical comparisons and find out which protein might explain some of the differences that we're seeing between experimental groups. And these tests that I will talk about are things that you heard about yesterday as well. So hopefully they're familiar to you a little bit from that, if not from prior. What we like to do, especially for proteomics, where we have 40% or more missing values, is both a quantitative and a qualitative test. So that quantitative test is going to tell us, are there differences in the average abundance of each protein between our treatment groups? And that can be done with an ANOVA or a t-test if you have two groups. And then we will also do a qualitative test or a presence-absence test, where we can see if there are differences between just the presence or an absence of a biomolecule between groups. So if you imagine here, right, in our table, we've got three proteins, A, B, and C. For protein A, we have observations in all 8 of our samples for our 2 groups. So we can do that quantitative comparison. Same with protein C. Even though we have some missing values, we still have enough observed values in each group that we can do that quantitative comparison. But protein B was not observed at all in the first group of samples, and it was observed in every one of the 4 group 2 samples. So we can't do that quantitative comparison, but there's likely something biologically interesting happening here with this protein, and a reason that it's not seen in group 1. And so that quantitative test can give us a p-value for that comparison based on presence and absence. If we were only doing that quantitative test, that ANOVA, we wouldn't be able to keep protein B in our data set, because if we tried to run an ANOVA in R, we would get an error back about all these missing values. So for the ANOVA, what we're doing is estimating a fold change for each group compared to another one for every molecule. And sure, you've all seen volcano plots before. What we've got here is our log2, in this case, fold change on the x-axis, and a negative log 10 of the p-value on the y. So each of these dots is a different protein. Proteins that are up higher are significant. And if they're out farther to either side, that means that fold change is larger. So these tend to be of the most interest when you get to the point of interpreting your data, which we'll hear about next talk. These plots are also used sometimes to sort of filter down, right, the number of proteins that you're looking at so that it's not overwhelming. If you have thousands that turn up significant, you may need to focus on a subset of fewer than 100, for instance, just so that it's manageable. For that qualitative test, showing this graph here again at the top, because of the large amount of missing values in proteomics data, and that's label-free proteomics. So here on the bottom right, we have 2 sample groups, A and B, 3 samples in each group are the columns. And the color indicates whether a protein was missing or observed. So the blue are the observed. So some of them are observed across all of these samples. Some of them are missing across all of these samples. And then some are sort of a mixture. And what we want to know is, are these patterns of missingness different from what we would expect just by random chance? If the data were missing randomly, then we want to know, is this any different, right? So if I see 3 missing versus 1 missing, should I expect that just randomly, or does that seem to be something significant that we should pay attention to? And I always like this set of graphs because it highlights the power of doing this combined test. So when we do both an ANOVA and a G test for a data set, what we tend to do is keep all of the results from the ANOVA that we can. So we could get a p-value for both ANOVA and a G test for the same protein. But if we have enough observations, get that quantitative result, then that's the one we're going to rely on. So these Venn diagrams are for 2 different data sets. So in one, I'm actually going to start on the right. In one, there were 102 proteins that were significant from the ANOVA, and there were 25 that were significant by the G test. So if we had only done the ANOVA, we would be missing out on these 25 proteins. On the left here, a different data set. The difference here is kind of astounding. There was a lot of missing values in this data. So there were 78 plus 17 that were significant by the ANOVA, and then over 400 that were significant by the G test. So in this case, having that presence absence test really added a lot of value because most of the proteins in this data set could not be analyzed using a quantitative method. And then I, of course, have to include a slide about multiple comparisons adjustment for p-values when we're working with omics data, because we have a lot of biomolecules. We might have a lot of comparisons that we're interested in making, depending on the experiment. So when we're thinking about p-value cutoffs, and we say that we want to call something significant if its p-value is smaller than 0.05, say, what that means is that we have a 5% false positive rate. So we're willing to say, I'm okay if 5% of what I'm seeing as significant is just by chance, and everything else is not. We're not going to know which are the 5% in our results, but that's sort of the typical cutoff that you probably see all over the place. The reason that we adjust p-values when we're doing multiple comparisons is that the more comparisons that we do, the more likely we are to have false positives. So there's two different ways you can adjust your p-values. So you can adjust because you are testing many biomolecules. So if you have 1,000 proteins and you're doing a test for every protein, then you can adjust using one of these methods here so that your type 1 error rate doesn't get inflated. Alternatively, or in addition, you can do an adjustment for the tests that you do for a single protein. So if you have 3 groups for one protein, what you're going to do is, if you're doing all pairwise comparisons, you'll have 3 comparisons for that protein. So you could adjust that protein's p-values for those 3 comparisons and do that for each protein. So there's 2 different ways to think about it. And for discovery studies, we typically don't adjust because we're just trying to see what those top most interesting proteins are. But if you're trying to do statistics for an exploratory study or something like that, then these adjustments become a lot more common. So just something to keep in mind, and consult with a statistician if you have questions, or the literature. This in particular, this topic, there's lots of information available. All right, that I will take any questions. David, I'll let you toss out if there's online questions for me.
I don't see any online questions regarding what you talked about right now. So is there any in the audience?
Okay.
Hi, so I had a question. Is there like a working list of different tools and methods available? Just because it's like my start of my proteomics journey, so I don't know what to use when to kind of build credibility in like the output that I get. So is there a place I can go to that has like, okay, these are what's available to you based on what you want to do, and this is what you should use because you are trying to get this. So just trying to get like a resource that's applicable, like I can refer back to it like every time I try to start something.
Right. Are you specifically interested in like tools for this type of work or for different parts of the process as well?
Different parts of the process as well. So like going through each process and figuring out, okay, based on what I'm trying to do, this might be more useful or less bias is introduced because of using this method. So
I'm sure there are. I'm thinking about sort of like annotation and identification, and that I don't know off the top of my head, other than I'm pretty sure that depending on who you ask, they'll have a favorite.
Okay.
For this type of data analysis, I would recommend looking at the PMART-R paper and seeing what is cited there. I am a little biased because I'm a developer of that, but we do really try to make sure that we're not providing methods without safeguards. So you can't just apply a function when you should have gone through a log transformation first, for instance, or done something about missing values. So trying to build those things in, and then on the sort of user interface side, if you're not interested in diving deep into programming, MAP is like the front end. Sort of, it contains an app that's the front end for that R package, and that also has sort of built-in safeguards. the tools that I mentioned on the slides too are other options, but may have different levels of those safeguards. So Metabo-Analyst and MS stats. Let me find this so that you can see. And then if David has anything to add, I'll let him pop it in the chat.
Okay, thank you.
Thank you.
Hi, I had a question about, you mentioned you'd have to filter out contaminants.
Mhm.
Has there ever been an instance in which, like if you're in like a discovery project, you didn't know how to figure out what the contaminants were? And kind of like, what did you do to try to like figure that out?
Yes, and I will say too, sometimes it's helpful to leave them in because they can sort of tell you, it can be kind of used as an extra quality control sanity check. I've worked on a metabolomics and lipidomics project in the past where we were obtaining samples from a larger consortium, different sampling sites. And when we got samples, we'd run them on the mass spec. I would get the data, process them, and we were trying to pull out any metabolites or lipids that were signatures of a disease. And we kept finding that there was one metabolite in particular that we kept finding as being like sometimes super abundant and sometimes not. And it took us a little while to finally figure it out. And it involved talking with the experimental scientists here, and then talking with all of the individual sites that were sending us samples. And what it ended up being is that there were two different types of sample tubes that were used. And one of them had EDTA in it, and one of them did not. And it was that EDTA that kept showing up really high in some samples, but not others. I'd say too, if you find samples that you're like, something seems off here, but I don't know what or why, like I always go back to the people who did either the processing of the samples on the instrument or the prep, or even the sort of originators of the samples, because sometimes they don't realize maybe that something was actually different enough for a sample to be able to be seen downstream. And so they might not mention it. And it's not anything that, they're not doing a great job. It's just doesn't seem like something that might be worth mentioning. And if you go back to them, they might be like, oh yeah, there was one sample that, blah, blah, blah. And they'll go back and look at their notes and they're like, yeah, actually this one, this tiny little thing was a little bit different. And you can kind of play detective there.
Early on, you had a slide about the high complexity of the system because there's so many different sort of modifications that can happen between the genome. Yeah. So to what extent are you able to predict some of these things like at the genome level so that when you see it at the proteome level or at the transcriptome level, so that when you see it later, you can say, oh, we expected this particular modification. Is there any way to do that, or is it just you go in pretty much blind?
A really good question. And I'm going to look at David and Paul and our other instructors to see if they have any insights.
Often we are blind, but if we do have previous knowledge, usually literature from some other techniques.
So like
rely on that heavily as like a gut check.
There's no like general pattern matching. You have to look at specific pathways and say, we see this pathway.
Sorry, can you repeat that?
There's no like pattern matching where you see like a general genomic signature for a phosphorylation. You can't do that. You just look for specific known pathways.
Right, those aren't coded in the genome. They're coded into kinases, but you don't know which kinases are being expressed or which are active at any given time until you do the proteomics.
We have some online questions filtering in. We have one where, can you generally explain when we do paired and unpaired statistical comparisons?
Or like when to do one versus the other? That's
Right.
the question. Okay. So this will be entirely dependent on the experimental design that you have. But one sort of classic example of when you would have a paired experiment is if you have before and after samples from the same organism. So this could be like a human serum sample. It could be some sort of like a soil sample before and after rewetting. If it's the soil, if it's a human example, maybe there's some sort of an intervention, like they're going to start eating something else or take a vitamin, something like that, right? But you have a sample from that same organism that you've got the initial, the before, and then you do something to it and you have an after. So those samples are more similar to each other than they would be to others from different after states. And so you want to do a paired analysis because you actually then are tying those together in the statistical framework, and that will give you more power to get your result. So ignoring that pairing is actually going to potentially show something as not being significant, even if it would be if you accounted for that pairing. So if you have paired data or correlated data is another way you might hear someone talk about it, you always want to account for that in your statistical model.
Thanks, Kelly. We have another question. If a data set doesn't completely satisfy the requirements of either a parametric or non-parametric test, what type of test would you suggest people use for non-normal or sparse data sets?
That's a good question. I'm trying to think of an answer that's not, it depends. Because I feel like it really depends on the scenario and what assumptions are not being met, right? You might be picking between the lesser of two evils, and it might be a toss-up. In general, I like to go with the simplest approach that is appropriate, and that can sometimes be a judgment call. So I would say if you're in a situation like that, consult a statistician who can help make sure that different assumptions and basically like parameters that you can play with in making that decision are accounted for.
Thank you. Are you tired yet? Because we have more.
I'll keep them coming.
All
We got
right.
time.
If the experimental design allows, is it fair to say that performing comparisons analysis between samples of the sample instead of samples across the batches can remove batch effects?
Can you repeat that, please?
Yes, I believe the question's asking that if you do each of the analysis between samples instead of groups, can that actually help remove batch effects? I think the answer is no, and you can talk about maybe Malbacher here.
Right. Yeah, I haven't seen that as an approach for removing batch effects. We've actually been studying batch effect correction methods, and Damon can talk more about that, either if you have questions online or during one of the networking breaks. But batch effect correction is still an evolving field of research, and there are quite a few methods that are available, but one of the things that's been missing is a way to compare them to each other. So how do you know for your data set that the batch effect correction method that you're applying is not making things worse or is even like doing anything? So similar to when I talked about data normalization, right? How do you know what the best method is? There's not that analogous heat map with the recommendation as this batch effect correction method is the best. But we have an R package that is available on GitHub that's tied to PMART-R that basically wraps up a number of the different batch effect correction methods that are available in the literature that we were able to incorporate into this R package, so that you can just sort of insert that batch effect correction step into your overall workflow without having to reformat your data to get it to the right form.